MTBR's tau's roles in Alzheimer's and its clinical implications. So, the background on Alzheimer's is that it affects over 25 million people in the world. It is a multi ecological disease with vascular risk factors as well as psychosocial factors. The physiology says that there is two stages. There's an amyloid stage with no clinical signs, and there is a tau protein stage, which then starts to represent cognitive deficits as well as cognitive impairment. There is no current biomarker to reflect brain tau pathology, and there is also no cure for Alzheimer's.
So as Yasmeen just mentioned, there is no cure and there's no way to assess the severity, which is why this research article's MTBR tau 243 finding of a potential biomarker is incredibly important because typically we have PET scans, but those are very expensive and inaccessible. So MTBR tau 243, if implemented as a biomarker, will make that much more accessible to a lot of patients. And as we know from the article, levels of MTBR tau 243 directly correlate with tau pathology, which is why it can act as a potential biomarker for detecting the severity of Alzheimer's.
In this particular slide, we are shown what a free tau monomer looks like, and a tau monomer that's actually bound to the microtubule of a neuron. And that's really important because when we look at tau, there is an MTBR region, which is the microtubule binding region. And that normally binds to the microtubule in the brain, and it usually stabilizes it when the tau is healthy. However, when tau is actually hyperphosphorylated, it becomes pathological, and then it actually causes the disintegration of the microtubule. And then what happens afterwards is that the tau monomers actually bind together in order to form tau tangles, which can eventually worsen the neural pathology.
So the mainstream article broad impact is that this serves as a new possible way of diagnosing Alzheimer's, assessing its severity, reducing the course of the severity of Alzheimer's if we can target the specific pathological MTBR tau species. And with further research, this could serve as a potential long-term cure, which is really exciting.
So in the research article, they found that tau species that contain the microtubule binding region are responsible for the formation of protein aggregates in the brain whose presence correlates with Alzheimer's disease severity. They found that specifically MTBR tau 243 concentrations correlated most with Alzheimer's disease severity and the disease progression. Okay, so they already have a way to assess clinical stages of Alzheimer's, and it's a 5-point scale called the CDR scale, and it goes from 0, which is normal, to 3, which is severe dementia. So they looked at those stages in relation to concentrations of different tau species, and they found that tau 243 correlated most with the progression of Alzheimer's.
And in this finding, we show that we can see the graphs of the concentration of the tau in this, the cerebral spinal fluid benchmark against the tau found by the PET scans. And the reason why the PET scans were actually utilized or used as a benchmark is because they're actually the clinical gold standard for measuring tau pathology in the brain. And as we can see from the 3 graphs here, we can see that the MTBR tau 243 had the highest correlation. And as such, we can see that it correlates most directly with the tau tangles in the brain.
And in order to determine all this, they first immunoprecipitated and chemically extracted their samples to find the specific proteins and molecules they were looking for. Then they ran it through mass spectrometry to quantify it. And lastly, to confirm all their findings, they did do PET scans.
And so the broad impact of our research, of the research article, was that the fact that MTBR tau 243 can be used potentially as a biomarker for measuring tau pathology and amyloid plaque in the brain of those who have Alzheimer's disease. And what's really interesting is that it can also be used for clinical staging of Alzheimer's, as well as tracking the efficacy of different therapeutics or treatments. And this could be done in clinic via a lumbar puncture, also known as a spinal tap, to see whether the therapeutics actually helped arrest the disease, improve the disease, or the disease got worse. And so pictured below are just two of the positions by which a lumbar puncture can be performed, either the lying position or the sitting position.
So when you're looking at both the mainstream article as well as the research article, they both have similarities and differences. The similarities include that there is the same information that is conveyed in both articles, and both articles also highlight the clinical implications of the findings. The differences include that the scientific article uses more precise and technical language, and it is written for those who are familiar with brain pathology. The mainstream article is written for more of the general public, and it summarizes the research article without data and detailed methodology.
So we'd just like to acknowledge Dr. Leah Michelle, Aaliyah, Washumat for this beautiful article, and our classmates for listening. And thank you. And if you have any questions, please feel free to put them in the chat.